Okay, this is part 6. Still looking at RNA viruses, look at single-stranded RNA viruses, and now we're going to look at the idea of the positive sense and the negative sense, or the plus strand and the minus strand version of single-stranded RNA viruses. So, a plus strand single-stranded RNA virus has a genome that functions like messenger RNA. In that, the genome, the RNA genome, has 3 letters of RNA, and those letters are codons, and those codons can be directly read by a ribosome and turned directly into proteins. One of the protein enzymes that will be encoded in our RNA genome will be the replicase enzyme itself. So somewhere on the virus's genome will be the gene that then makes the ribosome build the replicase. And then the replicase can go ahead and start to duplicate the genome. In this case, we start with our positive strand RNA, replicase builds the negative strand, i.e. the complementary strand, and then from the negative strand, the replicase then builds the positive strand once again. And it builds lots and lots and lots of positive strands, and all those positive strands get packaged up into new virus particles. When we have negative sense RNA viruses, what we have is that. So let's take a look at those strands. Here is, let's call this black one the positive strand. And here we can see 2 codons, AUG and CAC. And this can go directly into a ribosome, and it can be translated directly into a protein. You'll see if we have the complementary strand, let's call this one the negative sense strand, you can't send this directly into the same ribosome because it won't build the right protein. UAC is a different codon to AUG, which will call for a different amino acid. So the positive strand is set up for a ribosome to read it. The negative sense strand has to be turned into the positive sense strand. And then the positive strand can be turned into a protein. Now, our positive strand RNA viruses, one of the proteins they are going to build is replicase. Our negative sense strands, they can't build proteins just yet. So they can't build proteins out of the negative sense genome. So they have to bring replicase with them. And it's the replicase that they brought with them that allows them to generate the positive sense strand. And then the positive sense strand can be turned into proteins, which includes replicase. And it's this replicase that is packaged up into the new virus particle. Something else that is sometimes seen in single-stranded RNA viruses, be they positive or negative sense, is that some viruses, such as influenza A, have segmented genomes, which is just as it sounds. Influenza has 8 segments. You can see them represented as the orange dashes or the blue dashes. If a cell is infected with more than one version of the virus, as we see here, so we have our cell, it's infected by the green version of the virus, and it's infected by the red version of the virus. And as the genome gets replicated inside the cell, the various strands get all mixed up. And then when the virus tries to repackage, it'll repackage packets of strands that are different from each other. So we've had a rejumbling of the viral strands inside these new particles, and that can lead to large changes in the antigenic determinants of the virus. And this is known as antigenic shift. So here's our virus getting the green set. Sorry, here's our cell getting the green set and getting the red set. It's been infected by 2 viruses. Here we have the mixing and matching of the red and the green as they get replicated. And here we have the repackaging of the genome segments into new viruses. So we had antigenic drift before. Drift, slow, small changes. And now we have antigenic shift. Think about a shift. Sometimes big changes happening very suddenly. 2 different mechanisms that are causing the drift and the shift. Mutations building up in the genome, causing small changes to build up. A whole new version of a virus being generated all of a sudden as bits of 2 different viruses get mixed together. Antigenic shift, antigenic drift. Coronavirus is a positive sense RNA virus. It can be used directly as, the genome can be used directly as messenger RNA, and it can build proteins. One of those proteins will be replicase. Replicase is then packaged into the new virus particle. Replicase lacks that proofreading, so coronavirus is susceptible to antigenic drift. But coronavirus does not have a segmented genome, so antigenic shift is unlikely in a virus like COVID-19 or SARS-CoV-2.